Welcome to this presentation. Before we begin, I would like to introduce my YouTube channel, BL Nongsap, where you can access a wide range of educational resources.
The channel is organized into playlists covering pedagogy, ICT, training reports, internship materials, and many other resources designed to improve classroom practices and support the professional development of both teachers and learners.
I warmly invite you to subscribe and explore the content to enrich your academic and professional journey. Let's start. In this presentation, we explore Competency C 3.2 from NCFSE Science Education, which focuses on differentiating the characteristics of living organisms from non-living things for middle-stage learners.
Understanding Competency C 3.2.
Competency C 3.2 focuses on helping learners differentiate the characteristics of living organisms, including nutrition, growth and development, respiration, response to stimuli, reproduction, excretion, and cellular organization, from non-living things.
This competency forms a core foundation in biological science learning and is drawn directly from NCFSE 2023 and forms a core biological foundation for learners in grades 6, 7, and 8.
Why this competency matters.
Understanding life processes is a foundational concept for all biological sciences.
Through this competency, students develop critical thinking skills by observing, comparing, and classifying based on evidence.
It supports real-world applications, connecting classroom learning to health, environment, and everyday observations.
At the same time, it strengthens scientific inquiry skills, enabling systematic investigation and logical reasoning.
The NCFSE Linkage Framework.
NCFSE emphasizes that learning excellence emerges when all components work together.
Competencies define what students must know and be able to do.
Learning outcomes describe measurable skills and understanding.
Content focuses on the 7 characteristics of life and cellular basis.
Pedagogy highlights inquiry-based and hands-on strategies.
Assessment uses varied tools to measure understanding meaningfully.
The 7 characteristics of life.
To remember the characteristics of living organisms, the framework uses MRSGREN.
M, or movement, refers to the ability to move parts or the whole body in response to the environment.
R, or respiration, involves the release of energy from food, such as glucose, inside cells.
S, or sensitivity, means responding to stimuli like light, sound, touch, or chemicals.
G, or growth, is an increase in size and development over time.
R, or reproduction, is the production of offspring to continue the species.
E, or excretion, involves the removal of waste products from metabolic processes.
N, or nutrition, refers to obtaining and using food for energy and growth.
Learning outcomes for grade 6.
For knowledge and understanding, the learning outcomes are that Grade 6 learners list and identify the 7 characteristics of living organisms. They recognize that all living things are made of cells. They distinguish between living and non-living things based on observable features. They provide examples of each life characteristic from familiar organisms.
For application and skills, the learning outcomes are that students observe and classify specimens as living or non-living, providing appropriate reasoning. They design simple experiments to test life characteristics. They use microscopes to observe cellular structures in simple organisms. They record observations systematically in science journals.
For analysis and inquiry, the learning outcomes are that learners explain why viruses are debated as living or non-living. They compare different organisms that show similar life processes. They identify exceptions and discuss boundary cases. They formulate investigable questions related to life characteristics.
Learning outcomes for grade 7.
For knowledge and understanding, the learning outcomes are that Grade 7 learners explain the characteristics of life using biological mechanisms. They differentiate types of nutrition, understand the process of respiration, and describe sexual and asexual reproduction.
For application, skills, and critical thinking, the learning outcomes are that students conduct experiments related to life processes. They compare excretory systems across organisms, analyze responses to stimuli, create scientific models, and evaluate scientific claims using evidence.
Learning outcomes for Grade 8.
For knowledge and understanding, the learning outcomes are that grade 8 learners explain the molecular basis of life, understand cell theory, describe homeostasis, and connect life processes to evolution and natural selection.
For application and advanced skills, the learning outcomes are that students design complex experiments, analyze biochemical processes, use technology for scientific investigation, and construct integrated models of life systems.
For synthesis and evaluation, the learning outcomes are that learners synthesize life characteristics to explain survival and continuity. They evaluate claims about new or emerging life forms, apply concepts to astrobiology, and design original scientific investigations.
Pedagogical approaches for Competency C 3.2. The pedagogical approaches for Competency C 3.2 emphasize inquiry-based and experiential learning, fully aligned with NCFSE principles.
Inquiry-based learning. In inquiry-based learning, students engage in student-led investigations of living specimens. They test hypotheses related to the characteristics of life. Scientific questioning and problem-solving are continuously encouraged, enabling learners to construct understanding through evidence.
Hands-on experimentation. In hands-on experimentation, learners use microscopy to observe cells and organisms. They conduct experiments related to respiration, growth, and movement. Through these activities, students develop data collection and analysis skills, reinforcing conceptual understanding through practical experience.
Comparative analysis. In comparative analysis, students compare living and non-living things, study organism diversity, and analyze boundary cases such as viruses and seeds.
Collaborative learning. In collaborative learning, group projects, peer teaching, and debates strengthen communication skills, reasoning abilities, and teamwork.
Technology integration. Technology integration, like digital microscopy, simulations, and data logging tools, enhance observation, visualization, and accuracy.
Assessment procedures. Assessment follows a balanced and comprehensive approach, combining formative, performance-based, project-based, and summative methods.
Formative assessment. Formative assessment supports learning during instruction. Teachers use quick quizzes on life characteristics. They also use exit tickets after experiments to check students' understanding. Concept mapping activities are assessed to help reveal conceptual connections. Peer assessment is employed to encourage reflection and collaborative learning.
Performance tasks. Performance tasks, such as laboratory practical examinations, bridge abstract concepts with real-world applications by requiring students to actively apply their knowledge. When students design and conduct experiments, they demonstrate essential skills such as microscope operation, slide mounting, focusing, and structure identification, moving beyond purely theoretical understanding.
Project-based assessment. Project-based assessment, focused on MRS-GREN, allows students to develop a deep understanding of the 7 characteristics of life through active and hands-on learning. This assessment approach moves beyond rote memorization of biological concepts by engaging students in creating, analyzing, and constructing meaningful representations of knowledge.
Summative assessment. Summative assessment evaluates overall student achievement. Teachers conduct written tests that include multiple choice questions and essays. Practical examinations are used to assess experimental competence. Students' portfolios of experiments are also assessed. In addition, oral assessments are conducted to evaluate students' conceptual clarity and communication skills.
The following is the sample assessment rubric where teachers can use to assess learners.
Activity, living or not, classification challenge. This activity is designed for grades 6 to 8 and has a duration of 45 minutes. It is conducted in groups of 3 to 4 students.
The objective of the activity is for students to apply the MRSGREN framework to classify specimens and justify their reasoning using evidence.
Materials needed. Assorted specimens such as plant, animal, and fungi samples are provided. Non-living items include rocks, water, fire, and a robot toy. Edge cases include seeds, virus models, and coral. MRSGREN checklist sheets, magnifying glasses, and microscopes are used.
Procedure. First, the class reviews the MRSGREN characteristics together. Next, mystery specimens are presented in covered boxes. Groups observe each specimen systematically and complete the checklist to identify which characteristics are present. Specimens are then classified as living, non-living, or debatable. Groups present their findings with supporting evidence, followed by a class discussion on edge cases and reasoning. Finally, students reflect on what makes classification challenging.
Common misconceptions. The following misconceptions are commonly observed while teaching life characteristics. Addressing misconceptions is essential for building correct scientific understanding.
Misconception 1. Movement means locomotion only. A common misunderstanding is that movement refers only to visible locomotion. In reality, movement includes growth, internal processes, and responses to stimuli. Plants also show movement, such as bending toward light or opening and closing of flowers. To address this, teachers can show time-lapse videos of plant growth and tropisms and discuss movement at cellular and organism levels.
Misconception 2. Fire is alive because it grows, moves, and reproduces. Another misconception is that fire is alive. Fire lacks cellular organization, does not truly reproduce because it has no genetic material, and does not maintain homeostasis. Teachers can use comparison charts to emphasize that all characteristics of life must be present for something to be considered living.
Misconception 3. Viruses are definitely not alive. Students often assume viruses are clearly non-living. In reality, viruses show some life characteristics but require host cells to carry out life processes. This makes them a scientifically debated category. Teachers can use viruses as a case study for class debate, highlighting scientific uncertainty and evolving knowledge.
Misconception 4. Non-living things do not change. Another misconception is that non-living things do not change. Non-living things do change through physical and chemical processes, such as rusting or erosion, but not through biological processes. Teachers can compare physical, chemical, and biological changes to clarify this distinction.
Misconception 5. Seeds are not alive. Seeds are often thought to be non-living. In fact, seeds are alive but dormant, showing very slow respiration until conditions are suitable. Teachers can conduct germination experiments to demonstrate the difference between dormancy and active growth.
From competency to classroom. The following is the complete linkage framework, connecting all elements of teaching and learning. The competency requires students to differentiate living and non-living things using MRSGREN and cellular organization. Learning outcomes progress from identification in grade 6 to explanation of mechanisms in grade 7 to molecular analysis in grade 8. Content includes the 7 life characteristics, cellular organization, and comparative biology. Pedagogy relies on inquiry-based learning, hands-on experiments, collaborative learning, and technology integration. Assessment uses formative tools, performance tasks, projects, rubrics, and summative tests.
This video is prepared by Shri B.L. Nomsap, Diet Thadlaskin. Once again, make sure to share, like and subscribe and give your valuable positive feedback and comments. Thank you.